Good evening, everybody, and thanks for attending tonight's second session of our Chronic Wasting Disease webinar series brought to you by the University of Minnesota Center for Prion Research and Outreach, otherwise known as MinPro. I'm Mark Schwabenlander, and I'm the CWD Research and Outreach Manager for MinPro, and I'll be moderating tonight's webinar session. A few quick Zoom webinar housekeeping items. First of all, this session will be recorded and will be available at the MinPro website, minpro.umn.edu/outreach. The attendees' cameras and microphones are off during tonight's session. If you have any questions, please enter them in the Q&A tab at the bottom of the screen at any time during the presentation, and we'll cover those once Dr. Larson is done giving his presentation. If you have not interacted in Zoom, here are the steps for Q&A in Zoom. You follow along the green circle as I go through these. So first, click on the Q&A icon at the bottom of your screen. If you don't see that icon, just hover your mouse arrow over the bottom and it should appear. Next, click on the text box and type in your question. You have the opportunity to submit that question anonymously. So if you want to do that, click the little box next to Send Anonymously. And finally, you can click the send button and that question will come through to us. If you're listening to the session via telephone or watching the recording, any questions can be sent to our MinPro email, minpro@umn.edu, and we'll follow up with you. These questions will be addressed when the presenter is done tonight and will be anonymous if you send them during email if you're on telephone. So without further ado, tonight's presentation will be led by Dr. Peter Larson. He will be discussing CWD ecology and diagnostics. Peter is an assistant professor in the Department of Veterinary Biomedical Sciences in the College of Vet Med. Dr. Larson leads a diverse research program that focuses on the discovery of emerging infectious diseases, field-based molecular diagnostics, and the mechanisms of neurodegenerative diseases. Peter has over 18 years of research experience in molecular biology and genomics. Dr. Larson is leading a team at the U that is focused on the development of advanced diagnostic tools for the detection of chronic wasting disease, and he has organized a number of CWD educational and outreach events throughout the state of Minnesota, all aimed at sharing the latest science surrounding CWD research. So I'm going to unshare my screen. And Peter can start sharing his and begin.
Fantastic. Let me make sure we get this going here. How does that look? You see it?
Perfect. Yep.
Okay, great. Thanks so much for that introduction, Mark. And very happy to be here again for this next series or next lecture of the webinar series. This is similar intro slide that I used last time. This line drawing is from a book that was published by Teddy Roosevelt, second year of his presidency. And I'll be talking about the ecology of CWD today and be highlighting studies talking about the role of CWD prions in the environment. And I thought this was a perfect way to start to help visualize that. Throughout the talk today, and similar to the introduction I gave last time, be thinking about deer heritage. Also be thinking about deer behavior, deer natural history, right? How you visualize, how you see cervids in the environment, how cervids are interacting in the environment. So that understanding is going to play a role to a better understanding of CWD, the ecology of CWD. And then I'll also talk about some diagnostic tools, right? The current diagnostic tools for chronic wasting disease, some of the diagnostic tools that we currently have in our lab, and then talking about the future of CWD diagnostic tools. And I hope to leave plenty of time for questions. There's going to be a lot of things in here that you'll see that will open up a lot of questions about CWD in the environment. There's a lot that we know, there's a lot that we don't know. So there'll be a good Q&A session after this, I imagine. So just to review, last the last webinar, I covered the basic biology of CWD. And CWD belongs to this large family of prion diseases known as transmissible spongiform encephalopathies, or TSEs. All mammals have normal prion proteins. And I use this slinky example, right, as a three-dimensional object to better understand a normal prion. However, prions that become infectious take a misfolded form. Okay, this is that misfolded slinky, and I'm showing you in the protein diagrams in the lower right-hand corner. CWD can be transmitted through both direct and indirect routes, and we'll be talking about those indirect routes today. And just so everyone knows, you can go on to that the MinPro website, and you can go ahead and click that recording here button. That'll take you to a recording of last week's presentation. All right, so the outline for today's talk is I'll be highlighting the ecology of CWD. I really want to spend a little bit of time looking at lessons from scrapie. Okay, scrapie is a prion disease of sheep, and scrapie's been around for a long time. So a lot of what we know about CWD and the ecology of CWD actually is tied back to work for understanding prions and scrapie prions in the environment. Okay, and then I'll talk about highlight some papers, some studies that really do a nice job of showing how CWD prions interact with plants and soil and water. And then finish up with the diagnostic tools with first, second, and third diagnostic tool development. And I'll explain more about what that means. Okay. So again, this is so important to really hammer this home as we go into the downstream slides, because it's really important to have an understanding of prion biology when thinking about how prions can survive in the environment. So again, that left ribbon diagram, that's a normal prion protein. On the right there, that's the infectious misfolded protein. That's the form that can survive for a long time in the environment. Okay, not the normal form, not that ribbon diagram on the left there, but the one on the right, this misfolded beta sheet. This is protected, shielded, and it can survive in the environment for a long time. Okay, so that's what we're talking about when we're talking about prions in the environment. It's that misfolded form. Lessons from scrapie. I thought it'd be great just to start off with a story of scrapie in Iceland, because of really elegant experiments that go back to the 1940s, 50s, 60s that helped really develop a better understanding of how long prions can remain infectious in the environment. So scrapie was introduced by a ram. A ram from England was imported into Iceland in 1878. And then that ram turned out it had scrapie. And that ram was used for breeding efforts throughout Iceland. And the scrapie, just because of that, scrapie started to spread around the island. Okay. And that pink dot up here, this is where the ram was imported that had scrapie. And then these purple dots are where scrapie spread, okay, after that introduction. So in the 1940s, there was an effort to eradicate scrapie inside the geographic area of this red box here that I've drawn. Okay, so they went in and they eliminated all sheep from this area. And what they did is they have these large paddocks or large pens that had herds of sheep. And so they just went in and cleared all those out. So they took all the animals out. No sheep were present. And then after they did that, they waited a little, 3 years, right? They waited 3 years, and they restocked those paddocks with lambs. And what they found after a 3-year time was that the lambs over time began to develop scrapie. And there was no other animals that were introduced. These are pens, paddocks, large, fenced-in areas that had geographic barriers like glaciers. And really the only way that they could best explain this was that the scrapie prions were in the environment for longer than 3 years, right? For that three-year time. So the work out of Iceland, again, in the 40s, 50s, 60s showed that scrapie prion, that can remain infectious in the environment from anywhere from 3 to up to 16 years. Okay, long time for that misfolded scrapie prion. And I'm highlighting, you'll see I'll be showing some titles of different papers that tell this story. And Mark will be placing these papers on the website so you can go through and download a PDF and learn more about the research like that. But so the scrapie work is really, really pivotal to understand how long these misfolded prions survive in the environment. Remember, CWD wasn't identified until like the 1960s, 1970s, right? So CWD is a newer prion disease and cervix that came up. We've known about scrapie for a long time. So the work on scrapie, it's also, there's a ton of work on scrapie and looking at interactions with soil and whatnot, but there's some recent work that has shown that the misfolded scrapie prion can actually bind to dust and can be in dust inside barns, sheep barns that had scrapie. So you could actually set a device that would collect dust, and then you can go in and you can look and find the misfolded prions attached to dust particles. So it gives you an idea of how complex this problem is. Okay, and there's similar patterns that we're going to see when looking at CWD prions. Okay, shifting now to more CWD focus and the ecology of CWD. This figure comes from a review article that was published in 2017, and I'll kind of walk through this. So remember, if you attended the last session, I talked about direct transmission. So that's deer to deer. Okay, deer grooming each other, mating, bucks in the rut, you know, just herd, like just think about deer behavior and how close they congregate. That's CWD can be spread through saliva, through direct contact, okay? That contributes to most of the spread of CWD in the wild. But indirect transmission routes are also implicated. And what I mean by indirect is a CWD positive deer, and this is what I'm showing this figure here from the Zabel and Ortega publication, that CWD, that misfolded form, right, can be present in the feces, in the urine, can be present in the carcass of a CWD positive animal. And if the animal is CWD positive, and we know based on the data I discussed last time, that infection can occur, you know, 2-3 years. If an animal's positive for several years, they could be outputting prions in the environment. And this is what's shown in this figure here. So you'll have a CWD positive animal outputting prions in its urine and feces into the environment. That's what that lake is showing. It's also showing plants. Plants may be involved with uptake of prions, and I'll have a series of slides talking about that. Deer then CWD native deer can come along and be exposed to those misfolded prions in the environment, in maybe plants that have uptaken the prions, and then the cycle continues. So this is an indirect route of infection. So what evidence is there for this indirect routes? And we have to go back to some of these really pivotal experiments that were done in Colorado. So remember with this map, I was showing that the initial outbreak of CWD, or the earliest cases, were traced to a research facility in northern Colorado around Fort Collins. And so there's this great experiment where in Fort Collins in 1985, there were mule deer and elk, and they at a research facility, and they called those herds, right? They eliminated all those herds at a certain point in time in 1985. Those were positive, CWD positive animals, okay? The pins that they called that had the positive CWD positive animals in them, those pins were then treated with a bleaching powder to try to, I talked last time about bleach and how that may render the misfolded form non-infectious. So they're trying to render the misfolded form non-infectious inside these pins. And then they let it sit for a year. Elk herds were brought back in 86 and 91. And about 3 years after those reintroductions at this facility in Fort Collins, about 17 to 36% of those animals that were brought back that were brought in develop chronic wasting disease. So with an experiment like this, it's very similar to the Scrapie example where they culled everything, they took, animals out, they tried to treat the interior of the pens, they waited some amount of time, brought CWD negative animals in, and then after a period of time, all of a sudden they're developing CWD. So the best explanation for this was that those misfolded prions were present in the pens despite their remediation efforts. So there's a series of these experiments coming out of Colorado that are really important. In 2004, there was an experiment where they had uninfected mule deer separated into 3 groups. So group 1 was housed with deer that were positive. So they had mule deer housed with other mule deer that were positive. So offering, direct exposure. But then in a second group, they had CWD, they housed CWD negative animals with a positive carcass. Okay, so the CWD negative mule deer were housed in a pen that had a positive carcass in it. And I'm showing you this carcass in the upper right there, okay? So that's indirect. The only thing that was different between these 2 pens was that one had positive deer in it. The second one has a positive carcass in it. The third group, they had positive feces in there, okay? So the CWD negative animals, mule deer, were housed in a pen that had positive feces. What happened? Well, all 3 of those mule deer herds contracted CWD. Okay, so it's more evidence documenting that the misfolded prions present in the environment, that indirect contact resulted in CWD. Another experiment in 2009, bedding and water from CWD herds, positive herds, caused infections in CWD negative herds. Okay, so there's these sort of elegant, almost like challenge experiments that are done. Just really great work coming out of Colorado. And, I'm highlighting some of these papers here where they talk about those experiments. And all of this, it should give you a greater, as each study like this comes out, it gives us greater confidence that that misfolded form is the causative agent of the disease. There's other hypotheses that have been proposed, like maybe this is a virus, maybe it's a bacteria, but study after study like this is providing greater evidence, more evidence for the causative agent being a prion, similar to Scrapie, okay? All right, I'm gonna transition now and talk a little bit about some of the experiments that highlight the interaction between CWD prions and plant soil and water. So this figure, there's a lot of this paper caused a lot of buzz in the CWD research field. What this paper by Pritzko et al. In 2015 showed was the potential for plants to bring up prions through their roots, express them on their leaves, and that those or deposit them, on their leaves, and then those CWD prions deposited in the leaves could be infectious. So we'll sort of walk through this. So this is a wheatgrass experiment, and this is a paper that will also be on the website. They added CWD positive material to soil and I believe water, and then they grew up the wheatgrass in that soil that they used a brain homogen, and used CWD positive brain homogen, added it to soil, and they grew up the wheatgrass in that brain homogen. So what happened was they were able to detect the prions in the leaves using a technique called PMCA and Western blotting, which I'll highlight later on when I talk about diagnostics. But they're able to detect the misfolded prions in the leaves, and they fed those leaves to a hamster model. And unfortunately, the hamster ended up dying of a neurodegenerative disease. So they were able to show that the misfolded prion was taken by the plant, deposited in the leaves, did a challenge experiment by feeding the leaves to the hamster, and the hamster died. Okay, of a TSE. And so this is a really important paper that all of a sudden it highlighted this, the potential for misfolded prions to be uptaken by plants in the environment, and that may be a source of indirect transmission. So the route that this is occurring is that plants, they do uptake proteins as a nitrogen source, right? Well, this really obvious question comes up about, what about agriculture? What about, we know, thinking about deer behavior now on really across the Midwest, across this interface with of wild and agriculture, right? Think about looking out on alfalfa fields, maybe when snow is melting off in the spring, and you can see a herd of 100 deer on the corner of alfalfa field. Or think about herds of deer on the edge of soybean fields or in corn fields. And so that's a common component of the agricultural landscape, deer on agriculture. And because of that paper, and because plants have been implicated in indirect CWD transmission, in 2018, Norway actually banned the importation of hay and straw from CWD positive regions. So now when hay or straw are imported into Norway, it requires a letter from a vet that states that this material was harvested in an area that was CWD negative, or where CWD hadn't been identified yet. So already right now you have at least one country making a decision about what they're importing, just because of the potential interplay with CWD prions and plants. Recently, there was a case out of South Dakota of an elk farm that went positive, and they did all the trace outs, and they really tried to figure out, as with any farm that goes positive, South Dakota's equivalent of the Board of Animal Health went there and tried to understand, how this went positive. And what they came to was that there was hay bales that were being brought in from an area that had CWD positive animals in the wild, and on the alfalfa field there were CWD positive animals. And so they're hypothesizing that elk farm that went positive recently in South Dakota was, to their best of their knowledge, and if they understand all the things that were entering into that farm, their hypothesis is that it came from alfalfa. So that's a big question, is if you have a CWD positive herd in the landscape and they're putting out prions into the environment, let's say this is happening on an alfalfa field. I grew up on a farm, and I can tell you that first cut of alfalfa, that's pretty rough. You get a lot of soil in there, and it gets there's opportunities to get a hay bale that not only includes the alfalfa itself, but also soil and roots in there. And so it's a growing concern, right? Of materials being grown in CWD positive or endemic areas and then shipped to other areas. So we have, because of that, and there's a lot that we don't understand yet about how plants are intaking these, potentially incorporating these prions and depositing them in their leaves, like different species. Think about soybeans and versus grasses and native grasses. And so what we've done in our MinPro lab is we've assembled this fantastic team of about 10 experts, multidisciplinary, and we secured some funding to go ahead and start some experiments looking at CWD prion uptake in a variety of plants and commodities. So right now, and we're getting data like this week using some of the diagnostic tools I'll be talking about, where we're looking at barley and alfalfa and oats and trying to really understand how the prions are coming into those plants. Are they being expressed? Are they being deposited in different areas? Which areas? Why? And if you have a really good understanding of that, then you can develop ways to prevent it from happening, right? That's really important for agricultural commodities. Okay. Future experiments. So this right now we're focusing on commodities, but future experiments will be focusing on grasses and other commodities like corn and soybeans. But they're really, if you think about just how complex this issue is when you get out into the wild, right? Natural habitats, the ecology, the plant diversity in areas where deer are, across Minnesota, across Wisconsin, across Texas, it is very complex. So there's lots of unanswered questions, but, and really, I think what this boils down to is carcasses on the landscape. Like if you have a CWD positive animal that dies, it's very clear that that carcass is just a hot spot, right? A hot spot of the misfolded prions. And so the concern is with that really dense hot spot of the misfolded prions, that the plants there are an obvious, potential source for indirect transmission routes. And then along that same line, if you have a herd of CWD positive deer on the landscape, and then CW in the infection status is quite high, let's say something like 50% of animals in the herd are positive, and they herd up maybe in the winter, every day the feces, the urine that are coming out, right, that accumulates in the environment. And I'll talk a little bit about water, right, and how that plays a role into this. So be thinking about how prions can get concentrated in the environment. Soil. There is a ton of research looking at CWD prions in soil and some great work out there. And I don't have time to go into each one of these, but I just want to highlight, the fact that there's some just excellent work coming out of Jason Bartz's lab and work out of the Wisconsin lab, spacing, but Joel Peterson's lab, sorry, looking at CWD prions in mineral licks and natural soils. And just to highlight this, so soil, it can serve as a source for infection prions. We know a lot about this from Scrapie as well. Okay, again, so feces, urine, carcasses. And what the data that's emerging that's really important is that soil type matters. So you can have clay soils that can capture and enhance prion infectivity. There's publications talking about that. At the same time, you can have soils that are high in, it says humid, but humic acid, right, that can reduce infectivity. So you can have variation in your soil types that can either retain and really enhance infectivity, but you also have different soil types that can reduce. Okay, and I'm showing a soil map here of Minnesota, and it doesn't matter, there's a great soil maps no matter where you are in the US. That really matters. And so I hope that, as CWD spreads, that in each location, there are going to be folks that are trying to better understand the soil types to help with prediction of how CWD might spread or how those prions might interact with their local environmental conditions. So one thing we know about cervids is that they can ingest soil, right, both deliberately. So there's natural mineral licks where cervids will go to and really just take a bite out of the ground, if you will, and try to get minerals in. Okay, natural mineral licks. But also, and elk will do this too. They'll also consume soil during browsing, right? So CWD strain variation. I talked last time about strain variation, how there can be different three-dimensional shapes of the prion. That matters with how those prions interact with the different soil particles. So it gets very complex. Okay, but soil type does matter. For water, misfolded prions, they've been detected in water runoff downstream of CWD positive herds. So this is work, again, out of Colorado. There's a ton of CWD research out of Colorado, again, where CWD is hypothesized to originate from. But in these papers, there's work by Tracy Nichols et al. That showed they were able to detect the prions in snowmelt running off from endemic areas. Okay, so if you have a large herd and some percentage of those animals are positive and they're outputting prions, every day into the environment if that's occurring on snow, during the snow melt, what this paper shows is that they're able to catch some of that water and then identify the misfolded prions in their water samples. There's also work showing that they were able to identify those misfolded prions at the intake of a water treatment plant. Okay, so likely very low levels. If you think about prions coming into a water system, right, those are going to be diluted quite a bit, but nonetheless, they're able to detect them. There's also work showing that the more organic material in the water, the longer the prions remain infectious. Okay, so just organic material in the water, that's another factor of this interplay between those prions in the environment and water sources. So there's a bunch of unanswered questions about this. So CWD is relatively new in Minnesota. And as we know, in Minnesota, the land of 10,000 lakes, right? There's a lot of interest and need to identify potential pollutants in our waterways. So what we can hypothesize is if CWD is increasing in certain areas like southeastern Minnesota, I believe the infectivity rate is still around 1%. But if that were to increase or get to levels that are observed in other areas like in Wisconsin, higher infectivity rates, then it's likely that prions, these misfolded prions, will enter our waterways. Okay, and there's all sorts of questions like for CWD positive farms, do we need to be concerned about misfolded prions coming into, right, being washed off and getting into areas downstream of that farm? Does that then become a potential source of infection, indirect transmission to wild deer? Okay. At the same time, are farms that are downstream of a CWD positive herd, and maybe there's a creek that runs across the area, is that a potential route for indirect transmission? So lots of unanswered questions there, and the bottom line is more research is needed. So to summarize that, I know that there's going to be a lot of questions about this, the interplay between these CWD prions in the environment. But the bottom line is that those prions do not break down easily. Remember, they're shielded. So if you attended the first talk, that form makes it so that they're highly resistant to degradation. And the work from Scrapie and work coming out on CWD can show that they can remain infectious in the environment for years. So indirect transmission routes might include water, soil, and plants. And we really just need to have more research on to better understand that interplay between CWD strains, local environmental conditions, and infectivity. And our door alarm is going off, so I apologize for that. Okay, transitioning now to CWD diagnostics. Okay, this is an area that's of intense interest to everyone that's impacted by CWD. Hunters, right? Cervid farmers, regulatory agencies. There is a ton of interest in diagnostic tools, advancing diagnostic tools for CWD, because there's clear limitations with the ones that are currently available. Okay, so I'm going to split the current tests into 3 generations, and I'll explain more about what these mean. But if you view first-gen diagnostic tools for CWD, these are antibody-based. They're antibody-based, and there's a lot of, you've heard more about antibodies and diagnostics because of the COVID-19 pandemic. Well, the first-generation tools for CWD diagnostics are based on antibodies, and they need tissues from post-mortem, so harvested animals. Second-generation tools, they're quite interesting. They're based on prion amplification. So second-generation tools can actually leverage prion biology, how prions, how the normal and misfolded prions come together and induce a shape change. That's where second-generation tools reside in, is the prion amplification. You can use samples from both post-mortem and ante-mortem, so both harvested deer, but also live deer and environmental samples. Third-generation tools, this is an area that our research team is working on, where we want to take it to a next level. How can we leverage what we know about first and second generation tests, can we use maybe both antibodies and both in parallel with preon amplification to get to a really high sensitive and potentially portable CWD test, okay? That can be used for both post-mortem and ante-mortem samples. Okay, CWD first generation diagnostic tools. These are ELISA, Western blots, immunohistochemistry or IHC. And this kind of gets in the weeds a little bit, so I'll do the best to explain these tests. But ELISA, Western blots, immunohistochemistry, this is what's currently, these tests are currently used for CWD diagnostics. So here in Minnesota, if you are hunting in an area where the DNR wants you to submit your samples for CWD testing, they take those samples, they ship them. Right now they're being shipped to Colorado, and I think they're going to be going to Wisconsin here pretty soon, or for this next season. But when they go and they're going to be tested at a laboratory, they do this ELISA test, and then if there's a positive with ELISA, they follow up with IHC. And the main thing with these tests is that they use antibodies. All right, and I'll go into why that is. But these tests are the only approved, or these tests use very specific tissue types. So there's a reason, and I talked about this during the first talk, there's a reason why retropharyngeal lymph nodes are so important for testing, okay? And there's a reason why brainstem is so important for testing, and it's because the prions are enriched there. And some of you on this webinar may have pulled retropharyngeal lymph nodes. One thing that we've done in the MinPro lab, and this is work by Dr. Roxy Larson and Mark Schwabenlander, where they printed, 3D printed, this is a 3D printed white-tailed deer head, it's full size, it's all plastic, and you can see the anatomical features of the skull there. But if you peer down in here, and these pink tissues are salivary glands, a lot of times these are mistaken for lymph nodes. But if you peer down in there, you can see these two red nuggets. Those are the retropharyngeal lymph nodes. And so right now with these current first generation tests, and this is what the image in the lower right is showing, is you cut open, right, underneath, right behind the jaw there, and you have to dig down there and pull those lymph nodes out for testing. Okay, those are the retropharyngeal lymph nodes. All right, that's the primary tissue type because the prions get enriched there over time. Okay, so the way these first generation tests work, the way the ELISA and IHC tests work, there's actually an enzyme that they use. There's a chemical that they use that break down normal proteins. So remember the first talk, I said that there's both normal and abnormal prions, right? The antibodies that are being used right now can't differentiate between the two types. The antibody will bind to a normal just as effectively as it'll bind to an abnormal form. So it's there's a limitation for the current test. But to get around that, they add an enzyme to the sample. It breaks down all the normal proteins. The only thing that can survive is that misfolded prion, right? Because it can't easily degrade. So what this tests do is that you can actually then visualize the color change if the antibody is binding to the misfolded, to the prions that remain, the ones that weren't degraded by that enzyme. You can detect a color change. Okay, so in ELISA, it'll change a certain color, and then IHC, this is what I'm showing in the bottom panel there, is that they actually show up as pink dots. And this is how the current CWD tests are performed. They're using these antibodies and they're looking at color changes to determine whether or not those misfolded prions are present. Weaknesses, very time consuming. There are hunters that were waiting weeks for the results. I know last hunting season, and that's in large part because all these samples are going to central labs where they're processing like thousands of samples at a time. And so there's always just a lag time when you do that. But sometimes in great scenarios, you can get results within a few days. Worst case, it's 3 weeks out, right? So these tests can be very time consuming, require extensive laboratory expertise, big equipment footprint. So there's some equipment down in the lower right there that you need some pretty sophisticated equipment to do these tests when you're using you're trying to run through lots of samples. So this result, you need to have animals that were positive for some amount of time, like maybe 3, 4, 5 months. Like you need to have the prions really to start getting enriched in the lymph nodes in order to detect them with the color change. So there's a weakness there in that if it's an early infection, you might miss it using ELISA or IHC. A weakness is that these are really only good for, are best used right now for tissues from harvested animals. Antemortem testing is really limited based to just like rectal biopsies where they can do ELISA and IHC on. So right now, postmortem tests are the best. Okay, second generation diagnostic tests, PMCA, RT-Quick. I'm going to focus on RT-Quick because there's a lot of interest in this right now. These are seeded amplification methods. They leverage the way that prions misfold. And I used this slinky example right last time where you have normal, you have this abnormal form. If this abnormal form comes into contact and causes it to misfold, it induces it induces that shape change. These amplification methods leverage that, and I'm showing you that cartoon on the right. You can use this, the highly sensitive test, you can use it for lots of different tissue types, samples, biological samples. So blood, semen, urine, feces, even. You can use these methods on soil and in plants and water. Okay, so second generation tests are quite sensitive. I'm going to talk about RT-Quick and how this works here. RT-Quick came online. It's currently primarily used for research, probably within the last, I think, 6 years or something like that. But just in the last year or two, it's becoming more and more used for CWD diagnostics, like research purposes. It hasn't been validated yet, and I'll talk about that. But you start off with a sample. Let's say it's a blood sample. Okay. If it's CWD positive, you have these misfolded CWD prions present in that sample, that blood sample. You then take a small amount of that sample, it's blood in this case, and you put it into a well, a test tube that's full of normal formed prions. Okay, and I'm showing you there those ribbon diagrams. So you put it in a test tube that's just full of these normal prions. If the misfolded seed, if you will, if that is present, what happens is when you put it in the test tube and you incubate it and shake it, it causes all those other prions to start misfolding. It starts this chain reaction where they start misfolding. Okay, so in your test tube, you're leveraging what is actually happening in the animal when it has CWD, but you're doing it at a small scale inside of a test tube. And there's a way that you can detect that change. So you can detect that change by, there's a reagent, it's called thioflavin T. It is a molecule that binds to the misfolded prions. It binds to the beta sheets and the amyloid. It gets really complicated, but as that reaction happens and you have the thioflavin T there, and if the misfolded form is present, it starts to amplify and amplify. And you can detect that using a microplate reader. This I'm showing you in the lower right hand from our lab. This is a PhD student in my lab, Nancy Lee, who is using a microplate reader, just this box, that incubates and shakes. And then the curves I'm showing you from this paper in 2014, where that curve that comes up is when the amplification is happening and you get a lot of misfolded form, that molecule is binding, you can detect it, a light change basically. You can detect that using a signal, think of it like a laser, where it will change the wavelength, right? You can detect that saturation coming up, okay? And I'm showing you that these curves here in about 8 hours in this paper, they're able to detect a CWD positive sample. So that process, I mean, using a microplate reader, you can process around 46 samples at a time. Protocols exist for tissues and blood and feces. Results, it depends on the sample type. RT-Quick will work for a lymph node. You can detect positive samples within this 8 to 9 hour time frame. Using blood, using feces, it can be longer. It may be over a day using the current methods. But our Prion lab has about 4 microplate readers now, and we're routinely using RT-Quick pretty much every day now for research and diagnostic development. The method's currently being validated by the USDA, and that is going to take time. You know, I can't, someone from the USDA would have to speak to how long that method's going to take. And I shouldn't try to put down a timeline on that, but just know that it's happening, okay? So RT-QuIC is being validated by the USDA, and at some point in the future, hopefully it'll be an officially validated USDA test. Strengths can be used for lots of different biological samples, both antemortem and postmortem, as well as environmental samples. We're using RT-QuIC, and our initial results from plants is that we're able to use the method and identify the misfolded prions in the plant experiments that we're doing. It's consistent and as sensitive as those first generation tests. It's actually, it's more sensitive. It can pick up really early infections. Weakness, though, is that method, it still has extensive technical expertise that you need. You need a large lab footprint to do it. There's potential for false positives, right? Where the normal prions can just start to spontaneously misfold in the test tube, and that can be a false positive. And it can take a long time to complete. Okay, it can take over a day or up to 2 days, even depending on the sample type. Okay, so it's more sensitive, but it takes longer to complete. Third generation tests. This is what everyone is excited about and where we need to move with CWD testing. So I view third generation tests as the next phase. This is the future of CWD diagnostic tests. So the first tests were developed like in the 60s, 70s, right? The methods themselves, like ELISA, IHC. Second generation tests developed early 2000s with recent time. View those as like this old telescope that I'm showing you here. But third generation tests, and this is something that we're developing in our own lab, view it as this really high powered modern telescope, right? Where we're leveraging recent technologies. Technologies have been developed just in the last few years to try to create faster, cheaper, more sensitive tests, something that could be like taken out of the lab and into the field, okay? And there's several teams that are working on this throughout the world, and ours is one of them. And I talk a lot about portability and field-based assays, and the reason why I think this is an achievable goal of trying to get eventually a deer side test or something that could be done, set up at a DNR field station, is that in my own lab, I am using microfluidic and nano-based genomic technologies that are miniaturized that we're using for pathogen surveillance. These, I'm showing you the tools at the bottom here. They haven't been developed yet for proteins, but they've been developed for RNA and DNA pathogens like COVID-19. These technologies are really small. They fit in the palm of your hand. We have a DNA sequencer that's the size of a Snickers bar. And in the bottom there at the University of Minnesota sign, there's 2 suitcases there, Pelican cases. The entire mobile lab fits in those 2 cases. And we took that up to Itasca, and we screened for Lyme disease and ticks in Itasca at Itasca on site. And then we took the lab to Borneo. We went to Malaysia with it, and we generated genomic data in the jungles of Borneo. So this idea of field deployable diagnostic technologies, it's not new. We're using these right now for other things. The question is, how can we adapt these to something like CWD, right? How can we change that and use them for something like CWD, okay? The key is to have these large multidisciplinary teams. And this is something that we've done. So we have our diagnostic R&D team, and I can't go into the details of what we're developing, right? Because we have patents. We have about 3 or 4 provisional patents that we're submitting. We can't have public disclosures. But the key is if you bring in molecular biologists, experts in nanotechnology, engineering, antibody engineers, protein specialists, but also deer hunters, right? Cervid farmers, people that are on the ground, the end users. If you have all that together, and you can do some really fantastic things and develop these new tools, right? If you have all the players brought together in that team. So our research effort and for third generation diagnostic tests started in January. formally. It took us about 6 months to outfit our molecular lab and have all the equipment that we needed. We got it all set up in January. And despite the pandemic, we have now around 3 or 4 provisional patents that we're going to be submitting just in the next 3 weeks, 3 to 4 weeks. Okay. So we're making a lot of progress. And the goal of our work is to have functional prototype sometime in 2021 or 2022, the pandemic slowing things down, but have a prototype. Okay. Just showing proof of concept. There's a lot of work that needs to go to validation, time after that of reproducibility, right? But what we're doing is let's develop the technology now for a 3rd generation test to spark that and get it going. So I'm going over time, but the conclusions of this talk are that ecology of CWD is super complex, right? So you have CWD positive animals that are outputting prions into the environment. Where are those prions going? Are they being incorporated in plants? Are they being distributed in water? How are they interacting with soil? There's a lot of research going on right now to try to better understand that. Those misfolded prions can remain infectious in the environment for years, potentially. A lot of that depends on local conditions, right? We have extreme freeze thaws here in Minnesota. So the infectivity of the CWD prions in the environment, maybe in southeastern Minnesota, may be very different than what's going on in a place like Texas, for example, all right? So there's just a lot more research that needs to be done across the different ecologies and different environments that CWD occurs in. So the current gold standard tests, they're limited. Even the 2nd generation tests like RZ-Quick, they have their limitations. They're better, but they still have limitations, and we really have to work to develop 3rd generation tests. Okay, and that's the goal is to get to a field-based test. And based on the technology that we're already using in my lab, the fact that we can go anywhere in the planet and detect, viruses and bacteria in samples, I think we can do that for proteins. It's just going to take some time to figure it out. Again, I have to thank all the different funding sources. I want to open it up to questions. I know I went over, but Mark, let's go ahead and open this up for questions.
Thanks, Peter. Yeah, we have a few questions, and we'll stay around and answer questions as long as there's questions coming in. So the first questions are on the ecology area that we'll cover. And I'll start with the first question that talks really about the risk of CWD positive hotspots, and whether it's a farm or it's a hotspot in the wild that's been identified. What is the risk that, and should that be treated as a hazardous site? And what is the future of a spot like that, right?
Yeah, I mean, this is a great question, and it's, again, it obviously depends on the location. If it's a farm situation or in the wild, it depends on the number of animals in either situation, whether it's farmed or in the wild, the number of animals that are positive, how long was it there, all those factors that go into it. Think about prions being put in the environment. And so this idea of dose, we talked about this last time. What the paper that came out a couple months ago now showed was that a very small amount of prions, it's 300 nanograms of positive brain material, so was enough to cause and to successfully infect an animal. The question is, in the environment where, let's say, there's a carcass that was positive and there's grass around it, if an animal comes and browses all around there, are they getting that to that equivalent dose? Right? And we don't have a good understanding of that yet. So I would say that the question of the hotspot, it really depends on all those other factors of how many animals were positive, how long were they positive for, right? What are the best estimates for that? How long were they outputting CWD prions into that environment, right? And then what we're doing in our lab, like with RT Quick, is we want to go out right now to areas and collect water and soil and plants and see if we can find the misfolded prions. So work like that needs to be done to have a better understanding of what's happening out there.
Yeah, it leads into, I think you partially answered the question already, but infectivity limit of prions in soil, and what's the detection limit is in soil? Is it greater than the prions that or the infectivity level? And I think you
Yeah, so it's like we
that-
didn't. It gets tough because we didn't have a, these the papers getting at dose weren't. They're just coming out now, and then the tools to really get in and effectively look at environmental samples and prion content are just haven't been widely accessible, right? And so if you think about a place like a natural mineral lick where you have positive animals coming in, this comes from Joel Peterson's group out in Wisconsin. If you have positive animals coming into that natural mineral lick and they are, defecating, urinating, they are adding prions to that spot over time, that load is going to increase up. So the question is, what's that critical threshold, right? And what we need to do is get those samples. And if we're able to use, like with RT Quick, with the reaction goes, and we're able to see, how those curves come out, that tells us something about the number of prions there. And then we can also do other methods like the Western blots and antibody-based methods to try to have a better understanding. The other part of this too is, I should mention, you can do infectivity studies with rodent models. Okay, so you can have a colony of mice that you can take an environmental sample and inoculate the mouse, and will it develop, right, a TSE? Will it develop the equivalent of CWD over a period of months? And those are infectivity studies that people have done to show that the prions in the environment actually are infectious, right? So there's a lot of experiments that you can do to better understand that.
I guess similarly about dose and risk and environments is a question about, if a person was to walk through and eat berries in the woods, if they're CBD in a CBD positive area, is that a risk? And I think, maybe Corey will talk a little bit more about the risk to humans, but do you want to touch on that?
Right.
one?
So right now, and again, Corey's gonna talk about this next week, the best information that we have right now is that there's no evidence that CWD is causing disease in humans. There's concern because of strain variation, and Corey will talk about that next week. But the idea that you could go and consume, berries or what have you from the environment, I mean, the risk is super, super low or non-existent. Like it just, there's so many variables that go into that. And so the key is, where are those hot spots? If there's a blueberry plant next to a carcass, I probably would not, be eating that, but the risk is super, super low. So I don't, even though there are all this, there's data out there showing that these can be environmental contaminants, I don't want it to lead to sort of irrational fear of exposure, right? Because there's no evidence to suggest that there's any risk to humans at this point.
Okay, two mapping questions that I'm going to lump into one. One of them, it talks about the soil maps that you showed, and does that correlate? Do the soil types correlate at all to CWD positive areas in our state? And the other part of that is the distribution map of CBD. That's kind of spread around the state and the country that we talked about last time. Do we consider the spaces in between there potentially contaminated too? So one about the soil connection, and then what about the spaces between positives?
So I think, at this point, there's no evidence to suggest that the soil type is actually driving infectivity in Minnesota. It's more of when the wild population contracts CWD through whatever route, it's more of an issue of how those prions that are being output by those positive animals, about how, knowing what we know about soil, how long would they remain infectious? How are the soil types, in the herds that are positive in southeastern Minnesota? Are they clay or loamy? Or if they're clay enriched and positives have been identified in that area, then maybe there's a situation where prions at those hot spots remain infectious longer, right? Versus if CWD is in the wild up like in Crow Wing County, maybe there's different soil types there. I think it's more of, you have to look like long-term, right? Like if you look at if you think back to that map, and then getting to the second part of the question, if you think about where CWD has been endemic for like decades, right? And there's, herds where CWD infection may be quite high, and this is happening in Wisconsin too. Then at that situation, you want to, looking at the soil types will help with modeling. And this is something that we're doing too within the MinPro group, is that we're going to be doing some predictive modeling of if CWD were continue to spread in new areas of the state, 30 years out, 50 years out, like what do the soil types and how would they potentially interact with that, right? So more long-term modeling. The gaps in the US, like where CWD hasn't been identified yet, do we have a good understanding of what that landscape looks like as far as potential positives? Yes and no. I mean, it depends on the state monitoring efforts. It depends on, if animals look like they are clearly wasting away in the spot where CWD hasn't been identified at all, are those animals being tested, right? And so I think that all comes down to state level decisions and whether or not, right, animals that are potentially sick are being effectively tested for CWD. But the more, it's just like, think about COVID, the more testing that we're doing, the better the diagnostic tests that we have, the better understanding of where CWD is in the United States.
There's a question about disinfectants for prions. Are there effective disinfectants? And along with that, are there reasons why prions remain infectious for so long?
Yep. So about the remediation. So there are compounds that have been developed to help neutralize prions in the environment. There's some great work coming out of South Korea. So CWD was imported in South Korea by an animal that was transported, I think, from Canada, that was positive, ended up getting in South Korea, Surrey farms there. There's not a lot of land, right, in South Korea for these farming setups. And so what they developed was a method, I think with sodium chloride. They use mixtures of bleach. And what they do is they go in on these farms and they'll go and scrape out as much soil as they can and dispose of that in a landfill, but then treat the soil essentially with bleach and you want to change the pH. You want to try to change the pH of the soil, and if you do that successfully, you can render the prions non-infectious. So there are compounds that are being used by different groups to try to remediate at least farm situations. Now, the second part, what is it about these prions that make them so resistant to degradation? And we talk about that in the first webinar presentation. But what it is that you get the misfolding causes all these things. They're called beta sheets in the protein. They're just, think of them as just like rows and rows and rows, like stacked together, and they almost become impenetrable. And like they won't break down with normal methods. Okay, they're highly, highly resistant to breaking down. And so it's just that shape change at the level of the protein that's causing them to be resistant to degradation. Mm-hmm.
Talk a little bit about some of the diagnostic testing that you talked about. The question is, can you help a layperson understand what the implications and benefits of a field test give us versus the current diagnostics? So what's the benefit of?
Right. Great question. Why do we want to get this out, into, out of these, central labs and out into the field? And the answer is you want to have as near a real-time view of that CWD landscape as possible, right? You want to know, so for hunters to have to wait a long time to get results on their harvest, you have to maintain that carcass for a while, right? So if you're trying to decide whether or not you're going to process that for 3 weeks, that's not a good situation to be in for hunters. At the same time, for cervid farmers, right, it would be great if there was a way to rapidly detect whether or not there are CWD prions within the herd, right, on site. And so what we're working towards, right, is to get a field deployable test. And this can be in two ways. Like, one is if there's a DNR field station, we have a setup, something the size of a lunchbox that the DNR folks can use to test samples as they come in, and maybe have a few hours or even a day turnaround time at the level of the county or multiple stations within the county to test in real time. Then as soon as the animal is positive, if one's positive, one that the hunter knows, and also we can figure out where that gut pile is if we need to, and take take those materials off the landscape to prevent further infections. So the goal is to try to get to something that you have a much more rapid, reliable result, right? That's going to help reduce the spread of CWD. It's going to help prevent prions from entering our food supply.
Another question about diagnostics, and you mentioned the false positives with RT Quick because of the spontaneous folding. If that's possible during the test, why doesn't that occur naturally in the wild?
Oh, that's a great question. So the test it's a little bit different. So the test RT Quick, it can spontaneously misfold because that normal prion protein, the substrate, is actually it's a artificially produced hamster prion protein. So we can use bacteria and we can grow up these normal prions, a short segment of them, and use that as a substrate. Okay, so we can mass produce this, and just the properties of that that we're using can spontaneously misfold under certain conditions. Okay, so it's not the same molecule as what's happening, right? What you would find in the wild. Now, that said, spontaneous cases of prion diseases, they do happen. They happen in humans, they happen in cervids, but they're thought to be really rare, right? And so it's a great question that spontaneous conversion it in the wild, right, in natural animals, it can happen, but it's best we can tell is extremely rare.
All right, Peter, that covers our questions for today, and we're a little over the hour, but thanks for everybody for sticking around and asking such great questions. Reminder that the session was recorded and it will be available on the MinPro website, along with further information and resources to dig a little deeper. So if you missed last week's talk about the biology of basics of prion diseases, you can go to our MinPro website and take a look at that presentation, along with a bunch of other information that correlates with that first session. And the same thing will happen with this session and future sessions. Next week, we will be covering the human factor of CWD, and we'll have a guest lecturer, Corey Anderson from CIDRAP here at the University of Minnesota, where we'll talk some about the risk of CWD to humans and what you can do about CWD. So with that, thank you all for being here. Thanks to Peter for presenting tonight, and we'll see you next week.